Hello everyone, my name is Simon Rajendran. I'm one of the surgical registrars here in the Cork Cancer Research Centre in Ireland. The technique I'm showing you today involves the use of intact patient tissue for the study of gene delivery. Gene therapy offers exciting therapeutic approaches for the treatment of cancer. However, recent clinical trials have failed to replicate results found in animal models. The main reason I believe to be the variation in tissue physiology between animal and man, tumor heterogeneity, and differences in tumor phenotype. Ex vivo patient tissue model I'm demonstrating is the most stringent preclinical model available to date. This model involves slicing patient tissue into thin slices and maintaining these slices in vitro. Many gene delivery methods may be tested using this model. The viral vectors and DNA constructs I'm using express a firefly luciferase under the transcription control of the CMV promoter, allowing quantification of gene expression by bioluminescent imaging. Okay, let's go start this. The materials that are needed for this model are fresh human tissue, vibratome tissue slicing system, physical gene delivery methods to be assessed, an ultrasound machine, 6-plate tissue culture dish, 27-gauge insulin needle, non-toxic glue, gene delivery vector, which may be viral or non-viral, media containing serum, and culture media.